Good day. Today, I will be presenting our Thesis2 progress report entitled Antibacterial Activity of Endolichenic Fungi from Cladonia species collected from Papangan, Benguet, Northern Philippines against SRUs and E. coli. So, our group is composed of Katakutan, our group leader, Immaculangan, Ebora, Espinosa, Perez, and Redondo. Our thesis advisor is Mr. Lawrence B. Timbrez. So, for our executive summary, our research focuses on exploring the antibacterial potential of Endolichenic fungi, or ELF, isolated from Cladonia species collected in Kapangan, Benguet. We aim to test their activity against two standard bacterial strains, as Aureus, a gram-positive bacterium, and E. coli, a gram-negative bacterium. So far, we have completed several key milestones, including the acquisition of collection permits, specimen collection, and initial isolation of ELF from Cladonia species samples. Our next steps include molecular identification, extracting lichen metabolites, and antibacterial assays. So our study is guided by these objectives. First is to collect, characterize, and identify the lichen species from Cladonia species from Kapangan, Benguet using morphoanatomical analysis and talent spot test. Second is isolation and identification of the ELF from Cladonia species through molecular identification techniques. Third is to extract the secondary metabolites produced both by the lichen and by its Endolichenic fungi. Fourth is to test the antibacterial activity of both the lichen crude extract and the ELF crude extract against E. coli and SRUs using this diffusion assays. Finally, is to determine the minimum inhibitor concentration and minimum bactericidal concentration of these extracts using microdilution method. Okay, so for our work completed since last report. Well, since our last report, we have focused primarily on the subculturing and maintenance of our fungal isolates. On September 24, subculturing was led by Katakutan, followed by additional subculturing on September 29 by Redondo. On October 1, all members worked together on both subculturing and media preparation. And on October 6 and October 10, further subculturing and preparation of malt extract agar were carried out by Katakutan, Espinosa, and Redondo. Now for our data collected, well, as of the moment, our data includes the successful isolation of ELF fungi from Cladonia species samples. Each isolate has been replated and maintained on fresh MEA plates for purity testing. We have also documented the morphological characteristics of our isolates through visual observation and photographic records. The next phase will involve the molecular identification of these isolates through sequencing and phylogenetic analysis, which will strengthen the taxonomic accuracy of our data. So throughout our whole thesis progress, we have encountered several challenges. First is the contamination during isolation caused delays in obtaining pure fungal cultures. And to address this, we enhanced our aseptic techniques and implemented a stricter workflow within the laminar flow hood. Second, was the scheduling conflicts among members occasionally slowed down the subculturing process. We solved this by assigning dedicated time slots per member and maintaining a shared schedule for lab activities. Lastly, the university or laboratory closures due to unforeseen circumstances temporarily disrupted their workflow. And to mitigate this, we adopted a proactive replating schedule to ensure isolates remain viable during its downtime. Here we can see our updated Gantt chart. It reflects the adjustments made to our project timeline. So far, we have successfully completed the isolation phase and we are moving towards the molecular identification and metabolite extraction phase is scheduled for November and December 2025. The antibacterial activity testing is expected to follow in December 2025 to January 2026, while it will depend on sequencing turnaround time and extra preparation. So for our budget status, our original budget was 73,076 pesos. And out of this progress report, we have spent 39,920.85 centavos already, leaving a remaining balance of 33,155 and 15 cents. The expenses primarily covered culture media, reagents, consumables, and molecular analysis costs. Our budget remains on track, and we anticipate allocating the remaining funds for molecular sequencing and antibacterial testing materials in the upcoming months. So for our next steps, it will include morphological and talent characterization of Cladonia species in October, molecular identification of Endolichenic fungi, including sample shipment to Macrogen Korea for sequencing and subsequent phylogenetic reconstruction, which will last from November to December. The extraction of metabolites from both lichen and ELF samples will also be carried out from November to December. Then the screening of antibacterial activity from December 2025 to January 2026 to evaluate the inhibitory potential of the crude extract against E. coli and SRs. It will also be it will be on December to January 2026 rather. Okay, so for our team reflection, we have learned several important lessons throughout the stage. First is the importance of aseptic techniques. For even a small lapse can lead to contamination and significant delays on our thesis. Second would be clear communication among members. It is vital to discuss schedules early and reporting issues immediately so we can prevent conflicts and maintain the consistency of our progress. Finally, we learned that dedication and teamwork are key in managing time, task, and laboratory challenges effectively. All of these reflections have not only improved our workflow but also strengthened our collaboration as a research group, and we're doing good at the moment. That concludes our Thesis 2 progress report. Thank you for listening, and we look forward to presenting our final results in the next milestone.